Um, Hello everyone that's viewing this. My name is Jennifer Nix. I'm a double major in chemistry and biology. I'm a senior. I'm actually going to graduate in fall 2020. And I was thankfully selected to be a participant of the Summer Community of Scholars that is sponsored by the Honors College at UAH. I decided to do this project in cooperation with Dr. Love-Rutledge because I plan to get my master's in her lab after I graduate from undergrad at UAH, so just immediately starting it. And so this project was a little difficult to do, mostly because the COVID summer required me to be at school in order to use instruments and to actually prepare samples to analyze. But all in all, I think I was able to accomplish a good majority of what I was going to do regardless of COVID-19. So getting into it, the title of my project was basically, this is, so I actually presented to a group of high schoolers about this, but my project is called a validation of an MMR metabolomics method to assess blood-based changes in metabolism. And okay, I guess we'll get into it. So it's basically, I want to talk about how we normally process food. So when we ingest a meal, our body will convert that meal into usable energy. So that usable energy is glucose or sugar. Not to be confused with table sugar, the stuff that you would find in your pantry or at restaurants. That's sucrose, which is 1 part fructose and 1 part glucose. So our blood sugars will rise because of the meal. And because we have this heightened blood sugar levels, our pancreas, the organ that's like located right here, will start secreting insulin. And insulin is basically a hormone that tells other cells to uptake the extra sugar in our blood and store it for metabolic uses, such as rebuilding, destroying or recycling, or whatever our cells are going to do with that extra energy. And so this is a infographic that I took from the World Health Organization. My research is primarily on type 1 diabetics. For the general public's, for their simplicity's sake, they should only know about the 2 types. Type 1 diabetes is where the person cannot make their own insulin, so they require shots or however they get their insulin units in order to lower their blood sugar. And type 2 diabetes is where the person can still make insulin, but the insulin telling the cells to take up that extra sugar in their bloodstream doesn't work out so well because the cells just don't respond well to it at all. So they're in this chronic state of hyperglycemia. And the consequences that are listed here are basically the I have not been treated with insulin in a while, as in this is the worst of the worst. That could be stroke, blindness, heart attacks, kidney failures, or unfortunately amputation. And it's worth noting that diabetes is on the rise worldwide. 422 million adults have diabetes, with 1.5 million deaths caused by it. That's also 1 in every 11 people. So that gets me to the problem I wanted to address. So type 1 diabetes onset is unpredictable. So doctors and researchers cannot figure out the signs that someone might be developing type 1 diabetes. They can't run a panel of tests and be like, oh, you're totally missing this, or you're having more of this, so we're going to prevent you from getting type 1 diabetes by giving you this. There's no preventative for type 1 diabetes. There's just, oh, you have type 1 diabetes, and it's really unfortunate. Getting into more of the problem, type 1 diabetics will also develop other metabolic issues. So if you heard of diabetes, a symptom of diabetes is having a compromised immune system, that's one of those metabolic issues. The genetics of diabetes may cause detectable changes or metabolites that can be researched for prevention. So metabolites are basically just products of metabolism. You are producing metabolites right now. What keeps you alive is your metabolism. Why you're warm, why you sleep, why you breathe, that's metabolism. All of that requires energy from your cells. Anyway, and in order for us to investigate type 1 diabetics, our lab uses type 1 diabetic susceptible rats to study this disease. So I was provided these pictures by Dr. Love-Rutledge. I unfortunately was not in her lab to take these pictures, but they're so cute. These are the rats that she, I believe, took care. I believe it's 2018. I believe these rats were fostered in 2018. Unfortunately, they had to be sacrificed for science. So that leads me into talking about the two types of strains of rats that we have in the lab. So one is the LEW.1WR1 rat, which is a type 1 diabetic susceptible strain. It's well documented in the literature that they can spontaneously develop type 1 diabetes with a 2% chance. This means through natural means. And in order to encourage this process of developing type 1 diabetes, we inject them with a viral mimicking agent called poly-IC, also known as polyinosinic polycystidilic acid. I cannot pronounce that, please forgive me. For simplicity's sake, poly-IC. It's basically a double-stranded RNA virus that will attack the cells in the pancreas that make insulin units. And so because the poly-IC destroys all of the pancreas, well, not all the pancreas cells, the ones that make insulin, these rats will develop type 1 diabetes, approximately about 75% within 2 weeks. And then our other strain that we use mainly for control is the LEW.1WR1 rat. It is type 1 susceptible. So what did I do? So to recap, my research focuses on looking at the chemicals in the blood of type 1 diabetics, or when I finally can get to that step. These chemicals are side products of metabolism, or metabolites, and the artificial mixture itself was, so the recipe was made by a former master student that used to work in this lab. Her name was Genoa Collins. She made this mixture that was supposed to mimic blood or physiological conditions of type 1 diabetes, or type 1 diabetics. And in this mixture was 4 metabolites and a protein called bovine serum albumin. Bovine serum albumin is a very cheap protein, but the reason why we use this protein in particular is because basically all living things have this protein called albumin. Like even we have albumin. Albumin is a very sticky carrier protein that basically conveys all your metabolites across your body to whatever destination they need to go to. That's why we added it. BSA also is very difficult to work with MMR because it contributes to wider bands and noisier spectra, which is something we don't want in chemistry. But other than that, the 4 metabolites of interest that I was looking at that has been documented well in the literature for type 1 diabetics to have like abnormal levels of it, meaning they have too much or not enough, was aspartic acid, glutamic acid, glucose, and alanine. In order to look at these metabolites, special instrumentation was needed, and for me, it was nuclear magnetic resonance, or MR. So if you ever had an MRI done on you at the hospital, you've actually been around this similar kind of use of magnets. Although I don't think doctors are going around telling patients about how their instruments works and stuff. But anyway, so that leads me into talking about the instrument itself. This is the instrument that I used. It's the one at UAH. It belongs to the Department of Chemistry and it is overseen by my second mentor, Dr. Vogler. Shout out to him for teaching me how to use this thing. And that leads me into talking, in essence, in a nutshell, what MR is. So MR is a technique that uses magnets and radio frequency to see how a chemical reacts. How the chemical reacts with the frequency depends on the connections to all the atoms in the molecule. And lastly, MMR produces spectra that shows groups of atoms and their unique connections. So I want you to direct your attention to the spec that I have over here. This is an MMR spectra, MMR proton spectra. Proton meaning that we're only looking at the hydrogens. It's a relatively simple spectra of ethanol. I chose this for simplicity's sake because once we get bigger, it gets more complicated and convoluted, and I just want to keep it simple. So in a nutshell, I want you to look at the colors here. We have red, we have this red hydrogen, we have these 2 green ones, and we have these 3 blue ones. And so these hydrogens produce a unique signal that the instrument can see. And so we'll see the blue hydrogens here, the red hydrogen here, and the 2 hydrogens here. Why they're splitting like that, I recommend you look it up on your own time because that's a lot to explain. And now, this leads me into talking about my results, or what I was able to reproduce with her recipe. If you take a look at the left spectra, that is Genoa Collins, the former master student, her spectra that she put in her thesis. She produced this back in like May 2019, and she said she was able to get 75% yield from this, as in like from start to finish, from start as in making the artificial mixture and then putting it into the MMR machine. She was able to get 75% yield, which is pretty high and really nice. And if you, the one on the right is my spectra that I was able to make, which looks pretty darn identical, except for the fact that I got much lower yields than her, which is really sad and unfortunate. I think I still need to refine my technique, but the fact that my spectra is very identical gives me hope that I can refine my own technique and perhaps tweak a bit of her recipe in order to actually transfer these skills onto blood. So that leads me to the summary. Type 1 diabetics have altered metabolism. Our lab wants to use minimally invasive ways, such as blood and urine, to research type 1 diabetes, especially at onset. There's no preventative. There's nothing that stops people from getting type 1 diabetes, unfortunately. I used MMR to study type 1 diabetic unique chemicals in blood. Unique chemicals meaning that there's document in the literature that they have these abnormal levels of the metabolites that I used in my artificial mixture. And finally, MMR uses magnets and radio frequency to figure out what chemicals are present. And that should conclude my presentation. Thank you for listening. I'll see you later.